Pharmaceutical Processing, in conjunction with Interfex 2018, presents Interfex Live.
Well, you need to know that your system is still workable right now or can be easily upgraded. And what does that mean? Is it functionally filling BIOS for you? Do you have minimal interventions? Is it operating correctly and smoothly and efficiently? And if you have the basics running, even if it's a 20 or 25-year-old filling line, if it's still producing product for you reliably or can be upgraded with minimal effort, then a RABS retrofit might be the ideal thing for you. So we've done these RABS retrofits for all size facilities, from small to very, very large, high capacity lines. So you can certainly implement those without a problem. It's obviously a larger task, takes more time and more effort, but the overall project timeline is still the same. We typically do it where we do the preparation during shutdown in one of the in the beginning phase, and that's where we define the plan, we define a design, we do all the testing, and we get the operators involved in that, making sure we have a very good, well-designed system that's user-friendly as well. And then we go away, we build the equipment, and then during the next shutdown, we'll come in and actually install everything, do all the testing, and get you back up and running in really a few days. The previous methods definitely were very time intensive and took forever. And it was not so much even with the decon part of that phase with the gaseous decontamination systems, but it was also with the aeration phases. So the aeration is the removal of the sterilant or the decontaminating agent. And that's the thing that typically takes a very long time. Some people think that some of the earlier wet processes were actually pretty efficient because they weren't actually measuring how much residual gas got into the air. If you use peracetic acid, for example, and then you check that against the standards of today with an isolator system, you may have 3 to 4 to 10 times the amount of residue in the air that you would never find acceptable in an isolator system today. So the bar has been raised and it gets more challenging, but with the new technologies, we can do it much more efficiently. Now, with the new gassing processes that we're using both in large areas and rooms, as well as in isolator systems, we can turn these around much more efficiently and get our processes done in less than an hour sometimes, or at least a couple of hours, versus having to take an overnight or long sterilization phase. They do. The new strategies typically are getting better kill in shorter time period, and also we have better tools. Our company was concerned a lot about biological indicators. We decided to make our own because we were having some quality issues in the industry where biological indicators were clumping together and not effectively being able to be killed. That is not a fault of the hydrogen peroxide process. That's a fault of really the test method or the test equipment in this case. So if you have a well-controlled biological indicator, that can give you more reasonable and repeatable results. The other big technology change has come in the aeration. And our company has done some innovations with nanotechnology to have very high efficiency catalytic converters. And what's cool about the catalytic converters is they break down the hydrogen peroxide into a harmless water and oxygen mix. When I say water, I really mean water vapor. So it's not like it's wet all around. And it's environmentally friendly, and you can get rid of it much, much more quickly than you could in the past. With a typical sterilization cycle, you typically go through what we call a cycle development process. And you want to have a scientifically based cycle development process that isn't just guessing and making sure that you got kill on all of these biological indicators that you spread throughout the chamber. What you want to do is a scientifically based method where you show what the concentration is and the fact that it's been achieved and maintained during the proper exposure time, and then it's properly removed. When you do that, and then you go into your initial validation with your triplicate runs, then you get very reliable results. Going back a year later, you typically do annual requalification of those same cycles. And what you're trying to do there is just make sure that the system is performing as it was when it was originally installed. And that's typically done with single cycles, just to put a bunch of the biological indicators in those same positions that you checked them before. But you run a single cycle and make sure it's repeatable. And if you have a good biological indicator and you've controlled your process well, then the results are very repeatable. Well, with some of the new methods that SCAN is using, we have something we call SCANfog, which is really an atomization system of the hydrogen peroxide. What's great about that is not only are you getting faster kill and getting a direct access to the chamber in order to get that kill, but we typically use between 90 and 95% less peroxide than we do with the original systems. So that, combined with the nanotechnology in the catalyst removal, we're using less peroxide and we're needing to get rid of less peroxide, and it's a win-win situation. It gets you much more efficient cycles and much better results. So most of those technologies that you mentioned there are other delivery methods of hydrogen peroxide, most of which has been subsequently replaced with what we call SCANfog today, which was called, had other names in the past, and those are brand names of some of the products that we had. So we have a group within Switzerland that actually goes around in vans and can, have mobile decontamination units, and they can decon rooms for you as a service. And we do that throughout Switzerland and adjoining countries in that area. We do not do that in the United States, just because we're a much more spread out world here in the U.S., and it's not as cost effective to be able to, hop in a car and drive somewhere and bring everything with you. But the same technology is being used and applied to rooms that way now, and we're providing these pieces of equipment to customers who we can help them do all the validation work, and then they can have those for their own facility shutdowns or changeover procedures, and have all of those technologies available at their fingertips now for not only isolators, but for rooms.
Pharmaceutical Processing, in conjunction with Interfex 2018, presents Interfex Live.